Hi everyone, welcome to another episode of Whose Gene Is It Anyway? Show where we make up a bunch of really interesting DNA, do some bio stuff, design some things, talk about some things. I'm excited. I think it's going to be a fun day. Hope everyone's doing okay. Hope all is good and right in the world, or as well as it could be considering. Let's, as always, we're going to start by pulling an idea out of the hat. This is the hat. It's my plague doctor mask. Let's pull an idea. Let's see what we get, and that'll kind of set the theme of the show. There's a few in here. I say this every time, but there's a few in here that I'm like, man, I really wish I pulled that one today. All right, let's see what we got. You know what? This one says ivory yeast, but honestly, we've done so many yeast things. Kind of boring yeast things. I'm gonna save this one for another day. I'm not feeling it. Let's see what we got. Come on, something interesting. I'll take some votes on this one because I'm not sure if I'm feeling this one either, honestly. This one says grow butter. Sounds kind of gross. But yeah, let me think. So what do you think? Let's get some votes in the chat. Should we go with this one or should I pull another one? Meh. Okay, cool. I'm seeing throw it out. So throw it out it is. Pull a couple interesting ones. We'll see what we get. Yeah. Blah. I want something different. Like we've kind of had really basic ones for the last few. I mean the last one was fun because we got to do some plant stuff, which I enjoyed. Okay. Oh, okay. This one will be fun, I think. I'm liking this one. So this one says silica turtle. Silica turtle. So I take that to mean make a turtle with a silica shell, which sounds really cool. So what do you think? Should we better? I think. Yeah. Cool. We're going with that one. Silica turtle. I'm excited. Okay, so let me just update the stream name. There was another one in here that I hope I get soon. There's a couple CRISPR ones. There's a couple RNA interference ones. There's an optogenetic one in there, so I'm pretty stoked for some of those. Not that I'm not enjoying the enjoying all the yeast ones, because I am. I truly it's been really neat to go through some of the pathways. I'm kind of excited to do an animal one. We haven't done an animal one yet, and I mean this is going to be interesting because I've already got some ideas. So I think this would be fun. Cool. So before we get into that, there's a couple things I got to actually do today. First, I wanted to start with actually a little bit of show and tell and a story. So let's start with the show and tell first. I think I've mentioned this on a couple previous streams, but I've made a species of yeast produce beta-carotene. So beta-carotene is one of the dye molecules that's usually found in plants. I mean, obviously, carrots contain large quantities of it. It's not orange. It's a bright yellow compound. And when we eat beta-carotene, it gets turned into vitamin A, inside. So that's just kind of cool. So I found a paper and an iGEM project and a few other things where they had basically taken yeast and they made it express beta-carotene. And it's mostly contained within the yeast. Like, it doesn't really leak out because it's such a large molecule. But you could bake with this yeast, and that paper actually did. And they baked with the yeast, and they were able to detect like noticeable levels of beta carotene in the bread that they made. So that was pretty cool. So I'm going to do that. And so the exciting part is this is the yeast. So you can see just how yellow that is. Like, it is. Let's see if I can hold that closer so there's less glare. You can see how crazy yellow that is. Like, these yeast, when they're normal, like when they're not modified, look white. Like literally like pearl white. But yeah, so we've got some very, I mean, as you can see, like, this worked really well. So I'm going to be making some sourdough, and I think I'm going to open these up so you can get a look at the front, because underneath it just looks yellow, but some of the front, you can actually see some of the colonies look orange because they're actually accumulating one of the precursors in the pathway, which is red. So when you get red compound and yellow compound, you end up with orange colonies. So yeah, I can see some nice orange ones. Okay, so let's see if I can. So yeah, it's kind of hard to see on camera. This camera isn't super wonderful, but got some really cool yeast. So this is going to be one of the next videos. The video that I'm so there's going to be a video out tomorrow on my second channel, the Taste Emporium. Yeah, definitely. If you haven't already, go subscribe to that channel because it is going to be pretty good. We're going to be baking some bread. I actually have some of the bread right here. It's like eating clouds. It's wonderful. And I'm going to teach you how to do it with literally only like flour and salt and water. It's fantastic. So that'll be tomorrow. I'm editing that video currently. I'm just finishing it up, so that'll be up tomorrow. And so that'll be fun. One other little bit of interesting update news. I think I managed to get some of the DNA for the spider silk project into some yeast. With a couple caveats. Caveat number one. This is a little weird because I did the transformation like a week ago, and colonies only grew like yesterday, which I find very strange. Like, there shouldn't be that much of a lag, but who knows? Maybe there was. Caveat number two, it's the wrong species of yeast. I tried it just to see if I could even get the DNA into the species. It's actually the same species that I used for the keratin one, and it seems much easier to get DNA into than the actual species that the DNA was designed for, which is very frustrating. So I'm going to be moving forward on reworking that plasmid, and I'll show it. But once we switch to DNA view, I'll show you those really quick. I'll show you those changes quickly because I actually need to design a set of primers for PCR to basically just check these colonies and make sure that they're even real. Like, maybe it's just some weird contamination. So, yeah. So I gotta check those. In theory, I managed to get some like silk DNA into yeast, which is exciting. I'm going to refactor it so that it'll work in that species of yeast. And so that is actually progress. And then this week I'm going to be finishing up, fingers crossed, finishing up the E. coli version of the spider silk project. And then, it's the same thing I always say. Maybe I'll have silk soon, maybe I won't. And if I do, that video will finally be out. There'll be the lactose update next week. That video is almost done. So yeah, it's going to be bread tomorrow, lactose update next week, and then probably the keratin yeast after that, and then maybe the silk update. So that's going to be a lot of fun. Okay, let's jump into DNA view. I'm just going to make sure it's all good and ready. It is. You'll also see that there's a new donation bar for those interested in helping out. As much as I've been enjoying doing these streams, I would really love to be able to stream actually like doing some stuff, not just like designing DNA. So I want to get a capture card so that I can use my nice camera, and it should make these streams better as well. Like I've got a beautiful DSLR sitting right in front of me. So yeah, I'm going to get a capture card so that I can actually get the HDMI output from that, use the nicer camera, and ideally use 2 cameras at once, so that way I can have 2 different views, and then I'll actually be able to film some like working in the lab and stuff, which I think is I think that'd be fun. I think people would enjoy that. So with that out of the way, if anyone is feeling generous, you're welcome to kick in for that, and it's greatly appreciated. And yeah, so let's get into it. So we're going to need to make a new folder. I'm just going to do this first before we get into the spider silk stuff quickly. Glass turtle. Great. So this is actually going to be kind of fun because I kind of have done an enormous amount of research into biomineralization stuff. So hopefully I'll be able to find this. And this is definitely going to be one of those projects where I'm going to need everybody's help, because just finding all this stuff in real time is going to be very hard. So before we get into that, I need to design some primers, and I figured I would do it on stream so that you can see how this works. So these are all my different parts of the spider silk project. People were asking what's in the Frankenstein folder. That's what's in the Frankenstein folder. So yeah, you can see all the different versions. This is the version I made yesterday, and I can show that to you quickly. So basically, this is here. I wish there was a way to do single thing view. Okay, so let's zoom this guy in so you can see what's up. Fairly certain I deleted that, but anyways. So I'm actually going to have to zoom this in. So this plasmid, like this backbone, all the interesting stuff basically exists from here to here. The rest of this is just like backbone stuff, so I'm going to just kind of ignore it. Okay, so this is the gene. The AOX1 promoter and the alpha mating factor, the same thing that we've been using in previous episodes and have talked about pretty significantly. These both came in the backbone. We had a company stick this part in, so silk and then mineralization, and the mineralization is actually going to come in handy today, because this is going to do most of our work for us already, which is, I find, helpful. The several layers on the plasmid is basically just for organization sake, like, because this program wants to display as much of this text as possible, so it just kind of stacks them like this. But in reality, it's still just 2 things. Sometimes, and I haven't seen a plasmid that does this very often, but sometimes you'll see like one gene running in one direction, and then in like some other part of it, you'll actually have another protein that can be made if you read it backwards, which is really crazy. But it's pretty rare that you get to actually do that, because normally it would mess with your code. Gene Gun video will probably be a little while. Like I just had the idea for it, and also like Amazon is, as you can imagine, running pretty slowly. Like there's a month delay on everything. So even if I order all of the parts today, it'd still take a month to get here. So it'll be a while. But anyway, so we had a company put the mineralization and silk parts into this plasmid, right? And so this is built to work in Pichia pastoris. It's a type of yeast that I hate. But it was the only thing that the manufacturer had. Like they didn't have a plasmid built to work with Saccharomyces, which is what I prefer. Oh, we got somebody kicked in. I'm on the wrong tab, so I can't see. Oh, thank you, Alpha Centauri. Greatly appreciated. But yeah, so the company didn't have anything that did Saccharomyces, so we just got this figuring that, whatever, if it doesn't work quite the way that we want, we can just adjust it later. So this is the thing. So this plasmid already has a... I'll answer your question. Thank you, MD Roberts. I'll answer your question in just a second. I want to just finish this thought. So this version of the spider silk is actually something that hasn't. I mean, obviously it hasn't been made, but like, there's 2 additions that we're going to be making compared to the base thing that we ordered from the supplier. Thing number 1 is a GFP, so that the silk will be fluorescent. So that way, if the yeast make it all the way through this gene and successfully synthesize the entire silk, then they can make the GFP, and so then we'll know that the yeast are capable of actually making this whole thing. And the GFP is just kind of there. Like it shouldn't interfere with the silk itself, but it is it's there basically so we know if this worked from the get-go. One second. Okay, that was weird. Lost the cable. Anyway, so the other thing that we're going to be adding is this TEF1. It's a promoter that we've talked about many times on this stream already. Not today, but like in previous episodes. And this is a promoter specifically for Saccharomyces. So by adding this, what it should do is make this gene run in the easier to modify species of yeast, which is great because everybody has Saccharomyces. Pishy is hard to get and it's expensive, and it's just a shitty organism, whereas Saccharomyces is easy to work with. It transforms easily, it grows faster. It's fantastic. I love working with it. So this is the thing that we're going to be making. These little triangles here, these are primers. So we use a process called Gibson Assembly. So basically this part here, so TEF1 is a Saccharomyces promoter. So rather than paying a company to synthesize this for me, which would be less hassle but more expensive, because TEF1 is a natural Saccharomyces promoter, it should be fundamentally in every sample of baker's yeast you ever handle, which means that I should be able to actually isolate this gene directly from the yeast themselves, just using PCR. And so this half of the primer is what actually will do that. So it'll stick to the natural part of the, to the natural DNA. And so between that primer and then there's another one on the other end. So this marks the start and end. So this whole chunk should be basically copied out of the yeast. And then the other half of the primer is just there to match the sequence that you're going to be sticking this into. So in this case, it just matches there was a restriction site here, so you can basically make a hole that will fit this, and then use these weird primers to stick them together. So that's what I designed yesterday. We've actually already managed to do the PCR successfully for the GFP. So I literally have a tube of GFP DNA sitting in my freezer. And then we're just going to have to do the same thing with this, and then we should you mix all 3 pieces of DNA, add the right enzymes, heat them up to 50 degrees, and then immediately transform them into some bacteria or whatever. So yeah, that's exciting. That's what I designed yesterday. Just wanted to show that off really quick because I'm excited about it. Okay, so that's that one. Now, we actually need to open the original version, the unmodified, hasn't been messed with, basic. And so now you can see, you'll be able to actually see the difference. So this is the original version. Wait. What the hell? Why does this happen? That's not good. Huh. I accidentally edited this thing. I wonder if I can Control Z even between sessions. Oh, you totally can. Oh, that is so cool. I didn't know you could do that. So I've had this closed for, I mean, since yesterday. Like, I've had this whole page closed, but it keeps track of the steps that you took. So that's really cool. And I know that it worked because this is the restriction site that I was going to cut with. So you can see that there's now just the AOX1 promoter, and then it immediately goes to the alpha factor secretion signal. There's no TF thing here yet because we haven't added it, and there's no GFP on the end either. So now what I need to do is I need to design some really easy, basic, boring primers so that I can basically check to make sure that this piece of DNA is really there. So what I'm going to do is I'm going to make one primer. I want something that's about 500 long, just because that's a nice, easy size for a PCR product. It's big enough that it'll separate from any dimers, and it's relatively distinct, and it'll mean that it'll run through the gel pretty quickly. We got a question. So I've got to answer MD Roberts' question, and I want to answer this question quickly before we get back into this. So what does the mineralization gene do for the silk? So it's a weird. I'll click on it so you can see it. I don't think it's going to be labeled. Yeah, it's not. So there's several. This chunk. Wait, not this chunk. This chunk. This chunk is basically a series of short little proteins that are all stuck together that on contact with a mineral-laden solution will pull minerals out of the solution and in theory turn into solids. So like one of these is silica, which is what we're going to be using later for the glass turtle. One of them is calcium carbonate. One of them should stick to graphene or carbon nanotubes. And then the other one sticks to a metal. So the idea is that because this is stuck to the silk, if you were to ever take the silk, like say you made it into, like maybe you didn't weave it, like maybe you literally just got like a gob of the silk protein, you could stick it in, let's say, the ocean, and it will pull the minerals out of the water and solidify. So it'll turn into brick is the idea. Like the point with the spider silk project was never actually to make fiber. I don't really care about the fiber. It's a fun bonus, but like not why I'm doing it. The actual point is this. It's I want to grow bricks. So that sounds like it'll be a lot of fun. That's what the mineralization thing is. To answer MD Roberts' question really quickly, how long can results be kept viable in a Petri dish like you showed? I assume you're asking how long can you keep the organism happy on a Petri dish, and that really depends on the organism, the thickness of the agar. Like if you they'll last for a long time until they just dry out. There are some chances that it could maybe mutate, but if it's on a selection plate, which is what this is, like this has antibiotic in it, as long as you don't, as long as it doesn't dry out, like it's fine. And if it's starting to dry out, you just like take a little sample and streak it out on a new plate. But I mean, I've had plates in the fridge that remained viable for like a year. So, and yeah, the people are like, why can't you just buy bricks? That's not the point. One of these does calcium carbonate, which fundamentally requires carbon to be formed, and that means you would be capturing carbon whilst making bricks, is the point, amongst other things. And, you know, the easiest way to do this is, like, once you get this silk working, like, chuck it into some kombucha. Make, like, a weird fusion colony thing. So, like, it already has some structure to it, but now it just turns to rock when it hits water. Okay. Enough distractions. Let us make a thing. So we've got our secretion signal here. So I want a product that's about 500 long. I want half of it, and this was a suggestion from a friend of mine, but the idea is you make half of the product be in a chunk that is native to the plasmid backbone. So like the secretion factor was in there before the manufacturer messed with it. Like it's part of the normal thing. So you do half that is, here, and then you do another half in a chunk that they inserted, which is the silk in this case. So I'm going to see if I can basically make something that's about 500 long and goes, something like that. What's our length looking like? So I can go a little further in here. So that's about 500 long. I'm going to go a little shorter on this end and go a little longer on this end, I think. And a little longer on this end. I'm just kind of going back and forth. It doesn't need to be perfect. It doesn't really matter. I'm just kind of trying to eyeball this and see, like, okay, how far approximately do I need to go? I'm going to start with this end first, just so that I don't need to, like, just so I know how far into this we're going. Okay, so let's make a primer. So I always like to start or end my primers on C's and G's. They call this a GC clamp. We want something that's about 25 letters long, and we want a melting temperature of about 60 degrees. So that's actually good. My GC content, which is so these are the numbers I'm looking at down here. So we want something that melts at around 60 degrees, and whatever number we choose here has to be the same on the other end. We want as low of a GC content as physically possible. Ideally, definitely under 60, but like the problem is I'm trying to grab from inside the silk, which is inherently a bit of an issue because it is extremely GC dense. We can't see the numbers. Oh, the donations in the end, in the way. Let's see what this does. Well, do the thing. There we go. Is that better? Can you see it? I don't think so. I think it'll still probably be under the bar. But we'll see. Oh, no, I think you can... Yeah, I think you can see it now. Cool. So, these are the numbers I'm looking at. And I'm going to maybe grab a bit that's a little further over. I'm seeing lots of A's and C's, but less G. Well, the C's are going to... So, C and G basically make the DNA stick together harder than A's and T's. Like they're just inherently stickier. They have an X. So G's and C's form 3 bonds between them, whereas A's and T's form 2. So because of the extra bonds, it means more energy, i.e. more heat, is required to make the two things melt apart. So I'm gonna just grab a different spot. And we're gonna see if it's any better. Not to say that was bad inherently. I wanted a primer that was maybe a hair longer. That's actually pretty good right there. So that's 26. We got 50% GC content, and we got a melting point of about 60 degrees. So I would actually call that good. So we're gonna actually, you know what? I'm gonna do this in the primer menu. Just a little easier. So we're gonna make a pair of primers. We're gonna set this as the reverse because this is gonna be the end. Now we're gonna go all the way up to here, and I'd said I wanted to grab a spot right around here. So we've got a melting point of 59.5. So let's see how close. Okay, that'll do. Yeah, we got a G on the end. G's and C's, G's and C's. It's gonna be a little sticky, but that should be fine. So let's set that as forward. Cool beans. And in theory, that should be pretty good. The one thing I'm hesitant about, and so I'm going to actually exit out of full screen so I can open another tab. So there's this tool that Thermo Fisher has. It's the multiple primer analyzer, which is, so there can be issues when you make primers like this, where sometimes they'll stick to each other or they'll like bend back on themselves and stick to themselves. So you always have to make sure that they're fine. And so a tool like this is really good. So like it'll check. So if it doesn't find anything, then you're good. If it does find something, then you have a problem. And so it'll check both between the primers and on themselves. And by the looks of it, we've got, this is really important. So no self-dimers, no cross-dimers. So this means it doesn't stick to itself and it doesn't stick to the other one. This is very important. Then the last check that I'm going to do, and I don't think it's going to be a problem with the forward, but I do think it's going to be a problem with the reverse, is I'm just going to make sure that there's only... Oh, somebody kicked in. Well, thank you. So this is actually really good. I'm not seeing any other spots where this sticks. So I'm gonna cut it in half just because that's always the most likely to be a problem. This is good. So this doesn't stick anywhere that it's not supposed to, which is really, really good. And they don't stick to each other. They're the perfect melting point. Their GC content is pretty good, and they're a good length. So this means that we've got a really great set of primers, in theory. I mean, there's only one way to find out, but so we're going to call this Spider Brick. I think I'm on 19 and 20. It's exciting. We've moved up a decimal point. Or yeah, whatever. Cool beans. No. This one. Cool. So now we've got our primers made. So this was just a little bit of housekeeping that I had to get done, because I'm putting in a big order for primers. So I'm ordering this set. I'm ordering the set that I showed you before for adding the promoter here. So those are going to get ordered. And I'm also contemplating which set of primers I should buy for an experiment that I'm doing on testing for coronavirus. And the problem is I have a positive control. I just the primers that were in the paper that my friends were basing their test off of doesn't, like, it's not like that gene isn't in my positive control. So I need different primers, or I need a different positive control. And so I'm not really quite sure which way I'm going on that one. Okay. Okie dokie. I'm having a quick look at the chat because I've been doing this for a bit, so I haven't had a second to really look at it. Let's see what we got. Okay. I'm glad I can inspire. The biology's fun. It's just you got to have the constitution for it. It'll kick your ass 100 times, and you thank it for the honor. It's okay. So we're talking glass turtles. Okay, so now we can actually get into this. We're gonna need to do a couple of things, and this might actually be a CRISPR project. I'm not entirely sure yet. I need to think this through. Broken Bit says, Nausicaa in the Valley of the Wind was fucking amazing. I'm so glad that you watched it. It is a fantastic movie. So to anyone who hasn't seen that movie yet, it's Nausicaa of the Valley of the Wind. It's a Studio Ghibli movie. It's fantastic. Go watch it. Okay, so now we need to think about how we're going to make a glass turtle. Shell. I just wanted to see. No, some nonsense. Okay, so what I'm looking for, where there's two things that we need. Thing number one is we're gonna need something to localize to the shell. But you gotta think about, is such a thing even a thing? Does shell just count as bone? So let's learn a little bit about turtle shell. Because I don't know anything about turtle shell, and I would really like to. So let's see what is up. I mean, it looks like bone. It's highly so the turtle shell is a highly complicated shield for the ventral and dorsal parts of the turtle body. Okay, well that's not the bone of the shell. Okay, the bone of the shell consists of both skeletal and dermal bone. So innie and outie bone. Dermal armor. So, okay, that's really interesting. So it looks like it's just bone. So if you're going to mess with turtles, you'd need to mess with their bone, which sounds hard. Interesting. So the way I was thinking about this, wasn't really like a clear turtle. Like, I don't think that's possible, and it would die. I was more thinking about, like, what if you could give it like a extra crispy shell kind of thing. You know, like an igloo, like crunchy on the outside, squishy on the inside. Like, if you were to take the silica genes out of something like a diatom, like, could you make like a glass coating on the outside of a turtle? I don't know. So you gotta figure, like, is there? I mean, because there's like colorant and stuff. So is it keratin? Like, what's the actual outer shell? Let's look this up. Let's turtle shell composition. Let's see what's actually in it. So that'll give us a little more idea of what we're going for. Do turtles have shells? No. This seems right. Shell of the oldest true turtle. Okay, what's it? But what is it made of? Ooh, here we go. So it is a little bit keratin-y by the looks of this. It says beta keratins of the turtle shell. To me, that says it's at least partly keratin. So it's more close to nail. So let's see. Study for the first time sequences of 5 beta keratins from turtle epidermis. But is it like shell specific epidermis? Okay. So this just looks like keratin. This doesn't look like... Yeah, this is just like the keratin is the same as other things. Okay, well, that doesn't really help me. Interesting. I mean, we'd have to figure... So... We gotta figure out what's the composition of turtle shell, because if anyone's got a paper on the composition of turtle shell. Leave it in the chat. And someone yell at me if they post at it, so I see it. Okay, so Korwala says something really important. So turtle shells are made out of bones, and the exterior is covered with a keratin layer. So is there any way that we could, like, because I mean it's got a different pattern on it, so you got to figure that something is telling it to go, like, is controlling the genes on just the top half. So if you, I mean, this would be really, really hard, obviously. Like, this might not actually be something that's feasible to do in one design session. This might be a PhD project, but we're gonna see, just because I'm curious to know if anybody's actually done any of this research. So you got to figure, if there's a keratin layer on the outside, so skin basically. Yeah, it's the same keratin, it's just cells behave different because of the tissue. Yeah, right, but if something is triggering this color change here, right? So if something is triggering that color change in those specific areas, then in theory there's some promoter that you could find, or maybe it's a transposon, I don't know. I don't think it's a transposon because that's more of a plant thing than like a mammal thing. So with mammals, it's usually a weird hormone or like a, I don't know. There's a variety of different ways they can do it. So yeah, if anyone happens to know a shell skin specific promoter, which I feel like this is beyond, like way beyond the likelihood of anyone actually being able to just, you know, boop that up into the chat. But let's have a look. Coloration genes. Because I might be able to grab this off of another gene. That would make this easy. Because I mean, one option is to just make the, like, I'm hesitant to do. There's a simple / stupid way to do this, which is you just modify all of the keratin in the entire turtle to just do the silica thing, so it would just get progressively crunchier as it grows, but like you definitely kill the turtle. Like that's not okay. We're gonna keep looking around, because if there's no easy or obvious way to do this with turtles, then we might just have to do like a flower or something, like maybe like a glass flower could be interesting, because you could definitely do this with snails. I mean, that's actually let's see if maybe snails is the way to do this. Let's go. Snail shell mineralization. So with snails, and I mean definitely with like clams and stuff. We actually have a paper recommendation. I am stunned. 10.1093. The Malbev? Mal. SV. 265. Alright, let's see what comes up. Oh my god. If this is the thing that I need, I'm gonna lose it. Comparative genomic identifies epidermal proteins associated with the evolution of the turtle shell. God, I love the internet sometimes. Okay then. Whether the evolution, okay. Sequences and expression patterns of epidermal differentiating complex related genes. I am stunned. What? Okay, so the EDC comprises more than 100 genes, including at least 48 genes that encode proteins referred to as beta-caroteins. Or corneous... What's a corneous beta protein? That sounds like a bad joke. Yeah, I mean, there's another one that was suggested. The rapid evolution of beta-carotene genes contribute to phenotypic differences in turtles. So that'll be the next thing to look at if this fails. What did Karthik say? So what's a corneous protein? Oh, they're beta-carotene that somebody renamed just to be difficult. Okay, then. So let's see what we got here. Introduction. Blah, blah, blah. What do we got? So, what's this? I don't want to download the slide. I'd just like to see it. Oh, God. No, I don't want a PowerPoint. Okay, so these look like all the different keratins. What do we got here? Okay. Organization of skin genes in comparison to that of chickens. Okay. So this is chicken. This is turtle. Carapace formation. Okay. Extreme biases in amino acid composition. Okay, interesting. Do they actually list any of these genes? And also, like, it'd be good if they made like a map. That'd be really good. Oh, here we go. This seems useful. Oh, predominant expression site. Here we go. This is kind of exactly the thing that we need. So this one says shell, this one says shell, this one says shell. So EDQM. Don't know what that is. Beta A. So it must be a type of keratin. Try the supplementary data. Yeah, I'm gonna look at the supplementary in a minute. I just want to see. Ooh, it's a zip file. Oh, that is not what I wanted. I just wanted to open it. Alignment. That sounds interesting. Figures, tables. Let's try the tables. See what that does. Where is this? Oh, good lord, no. Open in Chrome, please. Why would it? No, it's just gonna stare. Oh well, I guess I gotta extract them all first. Extract evu. Tables is what we wanted. Open. There we go. Cool. All right, let's see what's in here. Oh, this is exactly what we wanted. That's very helpful, actually. Accession numbers. This is exactly what we want. It's gonna be a little hard to find the promoters in all of this, inevitably, but yeah, this is great. Cool. When are you planning on testing these or making a video? Like this plasmid? I have no intention of testing this. This is entirely an academic exercise. The milk and eggs plasmid I'm going to test because so that quote's going in today because I haven't seen anyone raise any issues on the GitHub file. So we're calling it good and we're putting in the order. And we haven't made any others that have been like, yeah, we should make that. I mean, like maybe the spicy tomato, because that sounded funny, but no. If we were gonna do the spicy tomato, honestly, there's other interesting ways to do spicy tomato. Like we could do like somatic fusion and like fuse a tomato and a pepper or something. Like that could be really interesting. But yeah, I don't know. Shrug. So this was the thing that we wanted. So EDQM or beta A, but it doesn't say which beta A is the one that we want necessarily. So, I mean, this is talking about, like, the evolution, but I would really like it to say, like, this one is the one that is in... Alright, so EDQM. Doesn't, I mean, so, let's look at this. Is there anything that says EDQM? M? Oh, yeah, there is. Wow, that is very poorly named. Epidermal differentiation protein containing a glutamine. Okay? So I'm going to look this guy up. So there looks like there's 6 or 8 of these. Yeah, there's 8. I don't know if there's any difference. EDQM1. Let's see if they've got it on Uniprot. They do. Because of course they do. European pond turtle. I guess we're working in European pond turtle. So that's a thing. Oh wow, it's really tiny. It's really, really tiny. Interesting. Let's see if we've got this on... Well, so they've got this. But it doesn't... Oh, EDQM. And here's the expression number. So, expression confirmed by RNA sequence data. So that's interesting. So we have that option as well. We can grab one that has like definitely been confirmed to be in the shell. So I think that's probably actually what we should do. So beta A and EDPCV. So EDPCV, that's these guys. Are any of them... This one maybe. Yeah, there we go. So this one has actually been confirmed by RNA expression in the shell, I think. So we're going to grab that guy, and we're going to see what's in it. Oh, it was a fragment? Okay. Oh, it's a fragment. Yeah, okay. Is this the same thing? Now, if I click on this, it should take me to the thing, right? Or is it going to be a jerk about this? Oh man, is everything just lagging now? Not this again. Oh good, now it's loading. It's loading, this loaded. In theory, we should have all of our stuff. Whole genome shotgun sequence. Cool. What is this? Dude. Oh boy. Okay. That's exciting. Oh, this is massive. Is this what is this? Is this the entire genome? This is the entire genome. That is not helpful. Bugger. Okay. Let's see if this comes up. I mean, if I know approximately... Oh, no, that's a hard no. Yeah, this seems to only exist in this... Like, it's in here somewhere, but finding it would be hard. Protein name in NCBI. Yeah. I will try that. Search. Nothing. Blast the peptide. The thing is, it wasn't... So if we go back to the supplemental, like you'll see that some of these say expression confirmed by RNA sequence data. So, I mean, does that mean that we could grab any of these and it should work? Because that's kind of what I'm thinking. Shell, beta O. So we got beta O, EDP-CV, beta A. So I mean, all this should be in the supplemental. I don't see... Oh, here's the beta. Beta O. Okay. But it doesn't... This one doesn't have the nice confirmed by RNA data. Yeah, it's weird. Like it comes with a number. Oh, look at this. They're all the same. It's all the same number. Oh wait, but it actually says where in the thing to look for. Okay. I can work with that. So if we go back, I should just be able to import this into Benchling. Search external database. Search. Nothing. It wants a different number. I mean, this should be the correct number. Why isn't this coming up? No, that's specified. This is frustrating. Is there any other ID on this? I mean, if we go back, it should. Yeah, there might be more ID stuff. Oh, get rid of the .1. Okay, I can do that. I mean, we'll see if it works. Search. It seems like everything's lagging again. Nope, no results. Rare. I mean, rarely. If it's an underscore, it should just be an underscore. I'm trying to see if there's any... Yeah, no, I mean, this is the... It should be the thing. It should just import. I don't know why it isn't. Maybe I just lose the NW. See if that's what it wants. No, it doesn't. It refuses. Well, that's significantly annoying. Manual input. Yeah, I know. I didn't want to. Is the problem. mean, yeah, trying to scroll through all this is just not going to happen. Is there a way to just download this? Alright, let's see what we do. Input raw sequence. Screw it. I'll rename this after. Pasty paste. Oh, it's not going to like this. Did it do anything? Paste. Oh, yay, it worked. I think I might have pasted it in twice by accident. But it was all highlighted, so it should have deleted the first copy. Okay, that's a thing. So now we need to go back to the supplemental, find, oh my god, base, is that 1,000,000? That's, yeah, 1,854,58, approximately. We're here, okay. So wait, it was 184, no, 18, yeah. We want, no wait, that's partial. So we want this one. We want number 7. So we want 183,697. Oh, too far. Oh, 1,837. Should be getting closer. Oh, I think I see it. 1,836. No, it was in the supplemental. 1,836,997. 36. Nine. Come on, we're almost there. 996, 9 there. It's around here. 9-ish. We've found the beginning. Oh, and it's an ATG. Oh, that literally could not be better. Wow, that is bang on. Okay. So it starts there. So now we just gotta find where it ends. So it goes 1,837,419. Somebody 1,837. 419, I believe. Yeah, 1,837,419. 1,837,46. 410. About here. How'd I do? Tag. Oh my, I did actually manage to just grab that whole thing. That's fantastic. So this is the, what's the name of this gene even? EDPC, this, whatever that. Boop. Save. Yay, we found it. Wow, that was actually easier than I thought it was going to be, if I'm being honest. Cool, so I don't really need this whole ridiculous thing. So let's just make a new... So it's called EDPCV7. So at the very least, we will have... I shouldn't have defined that as circular. I should have defined that as linear. Whoopsies. I have managed to copy 3 bases, like an idiot. Copy. Paste. Do thing. Did it do the thing? Now it did. Okay, cool. It's actually a really small gene. Honestly. Like, really, really small. This is interesting. So yeah, in theory, I need the upstream part, but the other, so I just wanted to grab this just so I had it, so I could always, like, if I lose it for some reason, I could just, like, copy it over. And the other thing that I'm thinking about is, what if we just modified this protein, right? Like, if this is good, like, we know this is in the outer shell part of the thing. So what we could do is, like, we could, I mean, there's been a lot of talk of, like, CRISPR stuff. Like, you could add it, like, you could, you know, we'd go back over to here, and you would end up making, like, a CRISPR thing to target, like, right here, or, I mean, maybe even do it, like, you know, somewhere in the gene, maybe, and you could add the little silica tag, so it'll just attract silica, so anywhere that this protein is expressed, you would end up getting silica, like, accumulating, essentially. So I'm not sure if I wanted to just grab, like, you could grab the whole upstream chunk of this and just, you know, if you grabbed a large enough chunk, in theory, you could probably grab a promoter. So but those are basically our options. Do we grab here and, you know, grab like this chunk here, for example, you know, some big chunky hunk of the thing, and then have it be, like, just use this and then have, like, a silica secreting gene after it and see if that does it. Because the other option is that maybe it's, well, no, I guess it wouldn't. I was thinking that maybe there's like a tag at the front of this protein that does something, but it's so tiny that I really don't see that being a thing. Also, that wouldn't really solve the why are you getting RNA expression of this in that area. So I'm not sure. What do you think? Should we go with modify this protein using CRISPR, or grab the promoter, and, you know, something that's promoter-ish, and then just have a silica gene? What do you think? The silica could misfold the protein. This is what I'm thinking. So maybe it is better to do the promoter thing. Not really sure. What would be cheaper to actually do? I'm gonna say it's about the same. Like, with if you're doing CRISPR, I mean, then turtles lay eggs. So actually, as these things go, that wouldn't be too bad to work with. It's not like it's not like modifying humans where the egg is so damn tiny that just working with it's a pain in the ass. What do we think? Promoter? Are people thinking promoter? Because I'm seeing that that seems to be the thing. The thoughts. Yeah, fuck it. Let's go promoter. I mean, it's nice that I've got this. So I have no idea how large this promoter is. So the nice thing is, for the silica part itself, it's very easy. But the promoter could be huge, it could be tiny. I have no real way of knowing that. So I'm going to just grab like 1,000 base pairs, and we're just going to see it. Oh, just replace the gene. We need the promoter and UTR for regulation. That is actually a good point. That is a very good point. So I'm actually going to have to grab, like, I mean, I have no idea how long the Terminator would be. I mean, I could just grab you know, I mean, let me just have a look. No, actually, the TEF promoter isn't very large. It's only about 400. So depending on where it's located, and they're always located pretty close to the actual gene. Like, there's no reason that it would be, like, way up here, and then there'd be a huge gap, and then there's a gene. Like, the promoters are usually pretty close. There might be enhancers that are further up that would, like, boost the level of expression or throttle the level of expression, but in theory, it should be pretty close. So it should be a matter of just, like, grabbing, like 600-700 worth of the front and 600-700 worth of the back, and then sticking a gene in the middle, and in theory that would work. I don't know why this has stopped scrolling, but here we are. Okay, we're back. Good. So I've got 1,000. I got 1,100 actually, so I'm gonna just shrink that down. I'm gonna grab an even 1,000. No particular reason, but just feeling it. I mean, if you really wanted to be like careful with this, I mean, what species of turtle is this anyway? This is the Chrysmer Murpidurber. I don't know. What do we got? Oh man, it's all the way at the top. Gotta scroll now. Let's see what little turtle we're looking with. This is a pond turtle, I think. Oh, that's so cute. Oh, look at it, it's so cute. Look at its little face. Isn't it adorable? It's so tiny. It's so wee. Oh, it's so cute. Okay, so we've got, this is apparently the turtle that we're working with. I'm so okay with this. I mean, obviously don't do this to the turtle. This is entirely an academic exercise. Don't modify the turtles. Glass Turtle's the name of my new band. Hilarious. So, cool. Very, very cool. So this is the turtle we're working with. That'll be fun. So I've got 1,000 base pairs in like upstream of the gene that we know is expressed in the skin on the outside of a turtle shell, which that sentence is entirely ridiculous. But cool. So we've got the gene. I'm going to just make a new DNA sequence. We're going to call this turtle shell promoter. Great. Man, this would be such a weird, like, thesis or like master's project. Come on, paste. There we go. So I have no idea which part of this is the actual promoter. In theory, the promoter is somewhere in this sequence. Don't know where, but this is just what we're going to be using. So then we need the terminator, which is going to be on the other end. So we're going to grab another, like, 500-600 base pairs and just assume that it's in there somewhere. Yeah, we're going to call it 600. Again, no particular reason. I'm just kind of looking and I'm seeing like this long, like lots of like T's and C and A's and stuff. So in theory, this is the right thing. Again, we're gonna make another DNA sequence. We're gonna call this turtle shell terminator. Great. I kind of want to blast this and just like, I mean, why not blast? Let's see what we got. I'm curious. I want to throw this chunk in and see if it's like if there's anything in there that's familiar. Okay. Let's go nucleotide blast. So I'm just kind of running this just to see what is up with this sequence because it's weird. And I want to know if there's anything that shows up as, like, this is a terminator. Because if there is, I mean, if there is, that's really wow. Jeez, that was fast. Interesting. Very interesting. It's seeing a little bit of... So, like, it's seeing the pond turtle that we were looking at before, which is interesting. But it's also seeing... There's like this chunk. I want to see this on graphic summary. That is fascinating. That is absolutely fascinating. There's like this weird chunk right at the end. So what BLAST does is you give it a sequence and then it searches through the entire database of NCBI, which is like this huge collection of genomes and amino acids and just like samples taken from organisms. What's really weird is this one chunk here. It says tRNA methyltransferase. So maybe this is actually spectrin. What is this? Spectrin repeat containing nuclear envelope family transcript variant. mRNA. This is weird. Yeah, like, and so you know, you give BLAST a sequence, and then it'll try and find similar things across organisms, basically. So what I did was I just took that chunk that we took out of the turtle genome, which does not appear to have pasted. Just very weird. Okay. So why did this... Okay, weird. Anyway, so we now have got something that we're going to call a terminator. We've got something that we're going to call a promoter, which is still very, very weird. I mean, there's a good chance that this doesn't work. But again, this is entirely an academic exercise. This is not, we're not really going for, you know, somebody should actually do this to a turtle. I don't know why anyone would do this to a turtle. Like, leave the fucking turtles alone. This is just fun. Blast the promoter. Yeah, I mean, that's a good plan. Should do that too. Promoter. Cool. Whoa, what? No. Escape. Cancel. I just want you to copy. What? Copy. Weird. Very weird. Okay, so there's definitely something. So, I mean, I probably grabbed something else with that terminator that's not the thing that we wanted. Nope. Nope, not what I wanted. Okay, we're going to paste in the promoter section. We're going to blast it. We're going to see what we get. I mean, I'm seeing all the same species. Same species, same species, same species. Ooh, different species. Okay, cool. Let's see the graphical summary. Interesting. So again, there's an area of similarity at the front. Weird. It'd be very strange that that's where the promoter is, though. But there is something. So I think this is actually, we would want to not include this. Personally, because like if this is showing up like not only across samples but like across species, yes, is ultra conserved element. That's very weird. It's a kinase. Yeah, to me this says you wouldn't want it. Like if we included this, it might run something bad. So maybe it's better to like shave, you know, 400 off the top kind of thing. So yeah, let's do that. Let's shave 400 off the top here, just because evidently there's something in there which we don't want. Hopefully I don't break anything. But again, we're not really gonna actually make this, so it doesn't really matter. Oh, come on, really? Cool. So we're gonna just delete that. This is now much shorter. Wonderful. The Terminator thing was somewhere like in the middle, so we're gonna shave about 300 or 400 off of that one as well, just because I don't trust it. I think it was around, yeah, it was around here-ish. So we're gonna call it at around the 300 mark, ish, and we're gonna just shave all this off. So now, in theory, we should have actually captured the promoter and terminator of that weird gene, of which we don't know what it does, nor do we care. So let's annotate. Oh, come on. Create annotation. This is going to be turtle shell terminator. Now we're going to do the same thing with the promoter, just so we know what this is. Because, I mean, I can't look at a string of letters and know what it is. So create annotation. This is going to be turtle shell promoter. Exciting. Save annotation. Cool beans. Now I need a plasmid. Do you have any suggestions for promoter recognition tools? Because I honestly don't know any, and I was just looking for something like that. No suggestions? Because if there is like an easy promoter recognition tool, I'd love to use it. So what is this? P-tub. This is the thing I was looking for. So this is what's called a mini circle. It's basically a really tiny bit of DNA that is reasonably easy to modify and is explicitly made to work in mammalian cells. Or animal cells. Anyways, I don't know if there's going to be any difference with. I mean, the only real difference with turtle should be that it's like, no, nothing. I mean, we got the promoter out of the turtle. It should work just fine. Targeting the TUB3 gene. Yeah, this is. I don't know if this is actually the thing we want. Background plasmid, knock-in donor. No. This one's fun. This is one of those things. Once you've done bio long enough, you start recognizing things. Like, if you're just scrolling past this, like you'd see this and you go, whatever, who cares? It's just a bunch of random genes. Except I recognize these as the Yamanaka factors, which are the like make your cells de-age, which is cool. CBS DTU CBS dot DTU DK promoter. Oh, cool. Let's see what this does. So this is just a tool that was suggested. Submit. Okay. Well, let's see what this does. I'm going to let it cook for a little bit while we keep looking for plasmids. I just want like an empty mammalian plasmid. Go add gene. So now we're just going to find something that we can, like, stick this gene into real easy. Oh, I think... Oh. And everything froze for a second. That was fun. Oh. Position 300. Wait, seriously? We actually found a promoter? That's... I am stunned. What? So in theory, this is actually a promoter. Am I reading this right? I've never used this tool before, so I don't really know what I'm looking at. I mean, has anyone used this tool before? Like. James, have you used this? Is this a thing? Because that would be really neat. From 300, not the start of your selection. Well, I mean, the start of my select. Oh, wait a second. I didn't. Whoops. from 300. So it should, what, is it 300 forward? This is prediction. Okay. Nope, Google found it. Okay, well, I'm going to go back in a new tab just to see what it tells me. Output format. Okay, highly likely prediction. Okay, so when position and position, okay, score. Is the prediction score for a transcription start site occurring within 100 base pairs upstream from that position? Okay. What in the fuck? Okay, well this is... Wait, did that say upstream? 100 base pairs upstream, so it's the other way, I think. Why would... What? So if it's 100 base pairs upstream, that means you start at 300. 300. And then 100 base pairs upstream, there would be a transcription start site. So 200. So it's saying that this bit is the promotery bit. Wait, somebody saw a Tata box? I don't see a Tata box. I mean, maybe I'm just like glazing over it because I'm just staring at letters. You know what? Screw it. I'm going to just say that somewhere in here is the promoter, so we're just going to include the entire thing. And this is the sort of thing that if somebody was doing a master's project on this, they could actually, you know, sequentially remove chunks and figure out which bit actually does the work. But I am not about it. So we're just going to grab the whole thing because I just don't care. I need just an empty backbone. Browse catalog. I want animal. I mean, there's not like an option for turtle, so we're gonna go with mammalian because screw it. It's you know, empty backbone. Let's see what we got. I mean, again, like, even if I try and look at this sequence, like, there is no way that I can look at it and go, yes, there is the promoter. Like, promoters are weird. So it's very, very difficult to be able to just look at it and go, like, yes, I know the useful part. Like, it's a string of letters. Like, human brain does not comprehend the way that this random string of letters is going to interact with thousands of proteins in a thing. So you kind of just got to, like, grab it and go for it. So I'm going to, like I said, I'm going to grab this whole chunk, and we're going to grab whatever's in the Terminator, and we're going to just, you know, start building a thing together. So, in fact, while I'm waiting for add gene, I'm going to make a new DNA sequence, and we're going to call this glass turtle. Great. And add gene looks like it's loaded, which is good. Yeah. What else do we got? So we got vector, fusion, and we want to find me, like, just do me the favor, find me a nice empty mammalian plasmid. Like, I basically just need, like, a multiple cloning site and, you know what, screw it. I can just find an empty, I mean, the nice thing is, because we're doing this with a, I mean, how would you actually do this modification? How do you select for the modified cells? I honestly don't know. When I say mammal, it's just animal, basically. I'm basically trying to think this through, right? So, a really basic mammalian, like mammal plasmid, right, basically has a few things in it. Do I have something that I can pull up really quickly? Technically, yes, I do. So this is like the most stripped down mammalian plasmid I can think of. So if I can see that in plasmid view, please. So basically, it's got like an origin of replication, an amp resistance site, and then just a mammalian promoter, and then whatever mammalian thing you want to make. In this case, it's a positive control for COVID. So really, all I need is an empty backbone. So I could use like PUC19, which would have this junk in it. So I could just grab that. If there's a multiple cloning site, I can just stick in the entire turtle gene, and this should just work fine. Interesting. I might just do that. Let's see what PUC19. I think that's the name of the vector I'm thinking of. Let's have a look. Let's see what's in it. Because I remember it being pretty stripped down. Like, there shouldn't be anything. I mean, there's a multiple cloning site with Lactase in it, but, like... Actually, that's not a terrible idea. We could do, like, a glass Lactase thing. So you could stain it really easily and see if it worked. Yeah. I mean, the alternative is you do this as a virus. Could do this as a viral plasmid. Unsure. Because, like, if you did it as a virus, then you could deliver it to the shell post growth. Like, you could deliver it to an adult versus trying to mess with the actual shell. And then you get more localized expression, but then it wouldn't be permanent. I don't know. Okay, yeah, you know what? Let's just, let's do something like this. But then there's an NDI site I could use. Is that one? Cloning site. Okay, yeah, let's just use this. This is nice and simple, and I can build on it, and it should be fine. So why did I close that? I needed that so I can download it. Open, please. There we go. Stop gene. Why is it always... What? Okay, good. No, I wasn't thinking virus from the beginning. I was just thinking. Okay, I can close this. I don't need this. I was just trying to think of the easiest delivery methods. So, if we go to glass turtle, should be able to import now. Should just be in downloads. I think this is, yeah, this is PUC19. This is perfect. Upload done. Close. Cool. So here's our multiple cloning site. So this is where we can actually like stick stuff in really easy. So we're probably going to use, what are our options here? We got Hin3, which is right nice at the beginning, which I appreciate. And it's kind of the furthest one over. Yeah, so we get a lac promoter. So this would actually kind of work in E. coli, which would be weird. Yeah, let's go with it. Let's go with Hin3. Do I have this one? Ooh, BAM HI. BAM HI. I like BAM HI. Primarily because I have some in my freezer. CTAG. Good. Let's do the promoter first. I'll just do this sequentially. So we're going to just copy it. Okay, so now we're actually into the phase of, like, building the thing. So so BAM-HI is a restriction enzyme. It's an enzyme that if you add, I've said this before, it's an enzyme that if you add it to this piece of DNA will cut right here. And I'm not seeing any other BAM-HI sites. I could check, but I just don't care. So we're going to, like, paste in the promoter. So come on. We're going to do a little pasty paste. So we've now added the promoter. I'm going to need to change all the annotations now. But turtle shell promoter is now in. So now if we go to the end of the turtle shell promoter, I'm going to go ahead and, so we're going to have to add the terminator further down, but that's okay. Now we need to deal with the actual silica-ish. So I would think that you would want, so now I'm looking for a secretion factor for mammalian cells. So let's see if we can find that. So mammalian, well, I guess it's not mammalian, it's turtle. Secretion signal sequence. Let's see what we got. So we're looking for the same sort of thing as the alpha mating factor that we've been using for yeast, but we need one for animals. I don't know if it'll necessarily work with turtles, but we'll see. So let's see what we're looking for. Baculovirus. We could have done baculovirus. That's true. I mean, I'm not used to baculovirus, so like I haven't really messed with it much. Mammalian cell culture, blah 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 blah. Transient expression, not helpful. So yeah, if anyone happens to know a plasmid that contains a secretion signal for animal cells, please post it in the chat. That would be greatly helpful. I mean, I'm looking for this, but we'll see what we got. Expression vector elements, signals, sequence selection, secretory peptides. Okay, cool. Yeah, fusion tags. This is what we're looking for. I mean, yeah, turtles are reptiles, but depending on what the secretion tag is, there's a chance that it would work in, in turtles. Because it depends what the protein is. If it's a protein that you also find in turtles, then it should work. Okay, do we have some, like, supplementary information? That would be really great. If there's, like, a supplementary thing, which I'm not seeing. No, so nobody got a secretion tag? Use of fusion chat. Okay. Fusion tags. Interesting. Not really what we want. Roller and secretion specific fusion tags. Okay, well that's not really helpful. I mean, reptile secretion peptide. Let's see what we got. Antimicrobial peptides in reptiles? That sounds entirely wrong. May modulate other aspects. No, that's not what I want. Definitely do not want snake venom. That's not right. Huh. Yeah, I mean, I'm not seeing anything particularly obvious. I mean, let's click on this one just to see what we've got. I mean, it just sounds so weird that I want to click on it and see what it is. I mean, antimicrobial peptides in reptiles. Can you imagine, like, going to the doctor and be like, you're going to need 20 cc's of reptile tears? Like, that just sounds so weird. And, like, if it was, like, you know, alternative medicine, it's definitely bullshit. But if a scientist says it, they're like, all right, maybe. Yeah, screw it. I'm going to go back to mammalian. I'm using mammalian just because it's been more studied, so the chances of it, somebody have found it already is much higher. Let's go with mammalian secretion tag. Peptide sequence. That sounds like the thing that we want. Ooh, ResearchGate. ResearchGate is helpful. Try a CD5 leader sequence. It works very well. Okay, wait. So can anyone suggest a leader peptide sequence for efficient secretion of a recombinant protein? CD5. Oh, you can use the interleukin stuff. And interleukin is in everything, so that would totally work. Oh, here's one. There are several signal peptides that can be used for this purpose. The one most used in our lab is the signal peptide CD33, which is this, because it's quite short and effective. I agree, that is quite short and effective, and I'm into it. Okay. Yeah, so do you attach some amino acid sequence to it that causes it to get transported outside? Yes, that's exactly what we're doing. So we're going to make a fusion protein. So it's going to be this stuck to a thing that does silica, basically. So we've already got our turtle shell promoter, so we're going to stick this in and see what happens. No, of course not. I mean, first of all, it only copied like a small amount of that, which is really, really useless. Reverse translate bio. Who's trolling? I mean, I could just do mods that are only testable, but like that's not the point. The point is to do something that's absolutely ridiculous and just go through the process of working our way through it. Like, I've never worked with turtles before. This has just been a really interesting this has just been a really interesting like academic exercise of like figuring out like how would you actually do something like this. So, oh no, this is the wrong thing. Wrong thing. Very much the wrong thing. But this should be the right thing. Reverse translate. Here we go. So we're going to put in the amino acid sequence. We want T's, and we want to make sure it all comes out on one line. Wonderful. Oh, and it's got an ATG. Great. Wait, this is wrong. That's an E. coli. That's not helpful. I'm going to go with mouse. Not for any particular reason other than, I don't know, mouse seems closer to the turtle, but it shouldn't really make a huge difference. Your talent and knowledge is being wasted on fantasy. I'm locked in my house and I need something entertaining to, like, both be entertaining and also distract me from the fact that the world is on fire. I'm quite enjoying this, actually. I do enough, like, serious bio as is. There's only so many projects that I can do. Each project is expensive, so doing things that are just wild and fun for absolutely no reason other than it just sounds cool is as much of a reason as I need to do something. So it's one of the reasons we're just gonna keep sticking with this. So we've got this should be our secretion tag, so we're gonna label this as such. So it's a CD33 secretion peptide. Cool. That's now in. Next up, we've got to add the actual mineralization chunk. I don't think that it would make it a transparent shell. We're basically going for just like making silica on the outside of the turtle shell. Like, see if it can have like a crunchy layer. So right now what we've got is a thing that should only be expressed. So because of this turtle shell promoter thing, this should only run, like this program should only run when it's in the shell of the turtle. So that's a really good place to start, I feel. Now we have to actually make silica, so that'll be fun. So we're going to be using a peptide called R5, which is a derivative of clam. Is it clam or is it oyster? I think it's either clam. No, wait. What the hell is R5 from? R5 silica. Let's okay. No, it's from Diatom. So it is actually Diatom derived. So it's the protein. The whole protein is called silifin, but only a very small part of it does the actual work. So in theory, I should actually have this in a folder somewhere, and it should be labeled. In theory. Otherwise, I got to open up Genome Compiler, which would be annoying. But, oh, maybe. Come on, be labeled. Labeled? Yay, it's labeled! Exciting. So we're going to grab this. This is the R5 peptide. It's what is currently in the silk plasmid. So yeah, we talked about doing CRISPR to replace one of the genes, but I wanted to do something that was plasmid based. I'm going to save CRISPR for a future thing. So anyways, we're going back to our turtle. So what we've got our secretion peptide. What I would really like is to add a string of Gs, so glycines, before we actually stick in the silica, just to make sure that there's no part of the peptide that is inhibited, because it really needs to not have anything on either end. Wait, do turtles have turtle immune system? No, not sea turtle, just regular turtle. So why do you want the secretion peptide? Because if you only have it inside of the shell, or like inside of the cell, you'll end up with like tiny little nodules of silica. Whereas I figure that if you have it secreted, silica should just build up in the whole area, is how I'm seeing this, basically. Immune response in turtles. I mean, they seem to do antibodies, I think. Yeah, okay. They've got antibodies, they've got immunoglobulins, so then the CD33 might actually work. How do you use plasmids on turtles? You would basically inject it into the egg. Okay, so, I was looking up the codon table. That was the thing. Codon table, please and thank you. Everything has an immune system, yes, but how similar it is to a mammalian immune system was the question. And because if it's not close enough, then this secretion tag won't work. All things have an immune system. You can't not have an immune system. Everything has an immune system. Even bacteria have an immune system. That's what CRISPR is. Oh yeah, so it's GG's. So this should be easy. So what I'm doing is I'm just gonna add what's called a peptide linker. It's basically just a string of glycine that is essentially innocuous. So we're gonna go GGA, GGU. Nope, that's a T. GGC. Is GGG also glycine? It should be. Yeah, it is. Cool. And then, oh shit, it didn't actually save. So GGA, GGT, GGC, GGG, GGA. So I'm just adding. That should be 5 glycines. So we're gonna label that as such. Glycine linker. Cool. Now I'm going to actually stick the R5 thing, which should still be copied. Yeah, it is. Wonderful. So now we have secretion tag, glycine linker, silica. What I'm going to actually do is I'm going to copy the glycine tag, stick it on the end, and then I'm going to copy the silica and stick a second copy. So that should be just nice. One thing that I could do, and I'm contemplating doing, is rather than this glycine linker in the middle, I could grab something that is keratin-like. So, I could actually grab... Where did it go? It should be in the glass turtle folder. So if I go back to the glass turtle folder, I could grab the whatever this is, and I could just modify it so that it's got a silica on either end. So it would be in the same spot, and it would just be a modified version of the protein. And I'm actually kind of into this idea because I don't know what this is. It was in the supplementary. It said what it was. EDPCV. EDPCV. Epidermal differentiation protein rich in proline. I'm actually not super into that. If there was like a really simple keratin, that would be good. There was that like fragment of keratin. What's it called? This is nice and short. Because beta O was the thing, right? So let's grab this accession number and see what we get. So what I think I'm going to do is I'm going to replace that internal linker with what should we call it? With a chunk of keratin. Oh no, that's a whole genome. That's not helpful. New plan. I'm going to grab the name of the species, and then I'm going to type in keratin and uniprot and see what we get. Keratin, uniprot. Because I don't think it matters which keratin. I just want something stretchy, basically. Come on, load. I mean, honestly, I could just grab a chunk of the spider silk and would do the same job. But, so yeah, Western painted turtle. Nope, that is human. I really was hoping for turtle. I don't know what this is. Let's find out. Oh, this is some garbage. This is not what we want. Let's just look up the species and see what they've got. Hemoglobin, sperm. No, none of this is really what we want. G protein. Yeah, none of this is really good. Spider silk is keratin. I mean, I don't know if spider silk is keratin. It's silk. It's different. It's a spideroin. It's sort of different. I mean, they're very similar, but I could just grab this. I don't know what it is though. I mean, here, let's have a. I want to know what this is. So let's create a forward translation just so I can see. Oh, I see what they mean by proline rich. I instantly understand that. So this is basically, they said epidermal proline rich something, and I mean you can see that it's all these P's. I think I'm just gonna grab a different keratin. I don't think it matters. Let's see what we got. Keratin associated protein, cytoskeletal, keratin associated protein. I mean, what are my species options here? Let's go taxonomy, maybe? Huh. That's weird. Carrots do not make your eyes work better. That was a myth started basically to explain to the Germans why, like, the British kept shooting down their planes and such. And the answer was radar, but they needed some excuse. I am seeing a lot of garbage, so I'm going to just go back. Keratin, uniprot. Let's see what that does. Alright, screw it. I'm just going to grab this. I really just need something that's going to be keratin-ish. Oh, Jesus. Okay, so that is actually just an enormous amount of glycine, is what that is. Okay, well, I'm going to just grab a chunk of this. Maybe not these aggressively glycine-rich areas. Although I don't know why I care. Shouldn't really matter. I'm gonna grab from the middle. I'm gonna just like grab this. And then we're gonna do our reverse translate again and just see what we get. Cool. So that's a thing. We're gonna go back to this guy, which I really need to rename. We're going to delete this because we have found an alternative linker. Actually, you know what? I'm gonna leave the glycine linker in there. Come on, go back, please. Oh, it's thinking. I mean, I could just use the really glycine-rich area. In fact, I'm gonna maybe nip back over to this, because if I use the really glycine-rich area, that sort of solves my I need a glycine linker, and also, actually, if I use this chunk, because having a huge amount of glycines stuck together like this means it'll be fairly crystalline, which means it should hydrogen bond to the rest of the keratin fairly well. Not sure what's going on here. Go back to here. Okay, I got silica. That's. If I go Control Z, it should get me a glycine linker back. And also jump around for no obvious reason. That's exciting. I'm gonna. Wait, did I do the reverse translate? No, I didn't. So I gotta do the reverse translate, grab the DNA that matches, pop back over to here, stick that in there. Grab this. Actually, I need to annotate this before I do anything else. Create annotation. Keratin fragment. Oh, I just closed it instead of saving it. That's exciting. Cool. Keratin fragment. And now I should have a Glycine Linker copied. I do. Wonderful. So now what this is going to do is this is going to have a... I can close this. So this should do... so we've got our turtle shell promoter. We've got our secretion tag, then a Glycine Linker, then a silica biomineralization peptide. Then we've got another Glycine Linker. We've got a keratin fragment, another Glycine Linker, and then another silica peptide, which is pretty dope. I'm going to throw another Glycine Linker on the end because this is actually going to be in... So this purple bar underneath is Lactase, it's lactase. So you could actually, in theory, stain this bit of skin, essentially, and with a compound called XGAL, and if it turns blue, then you know that this is actually being expressed properly. So basically what we've got is a weird keratin silica fusion protein. Yes, I realize this is really convoluted, but it's also BIOS, so that's just the name of the game. So we've got basically a weird silica keratin fusion protein also fused with Lactase, so you can know that it's there. And also, I'm going to turn that multiple cloning site off because I'm sick of looking at it. Delete, please. Cool. Also the Lactase, I should really... Where does this start? I mean, I want to kind of clean this up, but I don't really think it matters. So yeah, in theory, this should be a like if you were to actually like put this into Turtle, like you would actually in theory end up with like a functional thing. Like you should actually make a Diatom fused Turtle thing. The Lactase is overlapping just because the annotation hasn't updated. It just extended the thing when the reality is this whole chunk is not Lactase. The Lactase really just starts here. In fact, I can tidy that up. I can make that look nice. This ends at 1649, so we're gonna just open up our annotations, go over. Screw it, I'll just click on it. If I do that, I believe it was 1649 was what I said. Save annotation. There we go. Now it's just on the end where it's supposed to be. So we've got our turtle promoter, we've got our secretion tag, Glycine Linker, weird silica keratin fuckery, Lactase, and then the last thing that we got to do is first we got to add the terminator. That's important. But there's another thing that we got to do, which is add an SMAR sequence. Would we be able to add colors? I mean, maybe, but at that point, you're really stressing the ability for this thing to fold properly. Next, make turtles the size of diatoms. That would be very difficult. It'd be funny, but it'd be very difficult. So I'm gonna have. I already have this. So okay. So this next thing that I want to add is called an SMAR segment. So basically, what this will do is attach this piece of DNA to the wall of the nucleus of the cell, and this massively increases the amount of, like, the likelihood that this piece of DNA gets copied over as the cells divide. So with bacteria, we add this ORI, its origin of replication, so that this piece of DNA could get copied in E. coli. Mammalian or animal cells don't do that, so instead they need this thing called an SMAR sequence. Like, they don't have a known, like a simple known sequence that is copy from here. Instead, it's just whatever is stuck to the nuclear wall essentially does the thing. So I've got it in this plasmid, and I don't have it in this because there's nothing in here. That's really annoying. Bummer. I gotta hunt around on my computer for a second, so I'm gonna have to just switch this to this. Get to look at my face. Just because I need to be able to see my screen, and I don't need y'all seeing that. Or wait, is it even on this computer? That is the question, and the answer is no. It's not even on this computer. So, okay. Actually, you know what? I'm gonna leave it. I'm gonna leave it on that. I'm gonna leave it on this view for a second. I need to find this thing, and I don't know where I put it. So it's I know it's on my other laptop. So I'm just gonna pull this up for a second. I've got two. I'm using two different laptops. I don't have like a wonderful double screen setup. I'm in my kitchen. Okay, so I've got this stored. I have a folder of all my DNA files that I should have downloaded off of Genome Compiler, but there is a fairly real chance that I forgot to download this one, which, no, it's definitely here. I just didn't upload it to Benchling. So I'm gonna just pop open Benchling really quick, get this imported, and then we should be good to go. Okie dokie. Project, great. Yeah, this is really annoying. I really thought I'd loaded this in, but this should be fine. I mean, the nice thing is I know exactly where it is, so finding it wasn't hard, but it is annoying. I thought I'd transferred all of these, but I suppose it's good that I found out now. Cool. Okie dokie, that should have fixed it. So I'm going to just go ahead and set this back to scene, go back to here. Somebody just kick in? Oh, they did. Thank you, BP. Okie dokie. So we're back to the thing. I'm gonna have to refresh this, and that should fix it. I gotta say, this one's been interesting. I'm really glad we chose this one. Like, it was way more difficult than any of the other ones that we've done. So this has been really interesting. It's been a really good learning experience. So now if I go do this, yay, wonderful. So this is a little sneak peek of what you'll see next week when I do the lactase update. This is the version 2 of the lactase plasmid. It's real expensive. It's really, really expensive, which is why I have no intention of making it anytime soon. But I designed it so that I had the design and I can think about it for a while. So I should be able to go back to here now. I did copy the SMAR. I did. Yes, cool. Okay, so unfortunately there's not a restriction enzyme like right at the end here. But it needs to be read as RNA. I don't know how far in I can truncate this and have it still work. I'm going to need to make sure that I have the aminos so I know that I don't grab them in the wrong spot. What is going on? Why are you loading? Okay. So yeah, this is going to... I don't know if the catalytic end is going to get chewed up. It might. Okay. I'm gonna go ahead and assume that this is basically unnecessary. Well, I mean, it just needs to get read. I could put it but I can't put it at the end because I don't know where the terminator is, which means there could be a terminator in here somewhere. But if it's in turtles, it shouldn't matter. You know, this is kind of where I'm stuck. So I mean, you could cut further over. I don't know what. Well, actually, no. This is not really good. Cut. Let's do it. Let's do a quick digest. I just want to see if NDI cuts anywhere it's not supposed to. Nope, just one. No, that's a great cut. Okay, cool. So we're gonna cut here, I guess. So it's the A and the T get nipped off. So, boop, goodbye. Then we're going to paste in the SMAR. Cool. I mean, it's got a stop codon in it, so it should be fine. I'm going to add one right at the beginning, though. So TAG, TA, TAG. I always like to add extra stop codons just to, like, make sure. Oh, bummer. Okay, there's a frame shift in there. Nope, one more. There it is. Okay, cool. So we actually had to include this. So this is the extra junk that got added on that we needed. Added to a... Yeah. Cool, so now I know that I've added that. The SMAR is here. So basically the way the SMAR works, it needs to be read through a promoter. Wow, that is really large. I forget how large SMAR is sometimes and how tiny this was. Okay, so basically the way this works, right, is you've got our turtle shell promoter here. So the enzymes will grab onto this part and then scan through the DNA until they hit this ATG right here, where it basically tells the enzymes, like, protein starts here. Well, so okay, I'm actually out of order. What the promoter will do is the RNA transcriptase will grab onto somewhere in here. We're not entirely sure where. It doesn't really matter. And it'll just start transcribing RNA until it hits a terminator, which we still need to add. The terminator, it will make that enzyme let go. Throughout that process, it unwinds the DNA, and what the SMAR does, the thing we just added, what this does is when the DNA is unwound, various proteins inside the nucleus of the, like, cells of, in this case, a turtle, grab onto this sequence. And admittedly, this is a human SMAR, so I have no idea if this would work in turtles. I honestly have no idea if any of this would work. It's really just an excuse to talk about all these interesting, weird things. But so assuming that this is an SMAR that is even a little bit similar to what you would find in a turtle, and I'd have to, like, do some blast stuff to check that. Once this bit of DNA is unwound, so that the thing can get copied, various proteins would grab onto the SMAR and then bind it to the wall of the nucleus. It doesn't actually produce any protein. It's not a... I wonder if I can get rid of the translation, because it's not a protein. I honestly don't know. Whatever. It doesn't really matter. Oh, thank you. Thank you very much for kicking in. Greatly appreciated. So in theory, the SMAR would bind this to the nucleus and make sure that this whole piece of DNA gets copied. I don't know if it would. It's a turtle. It's meant for a human. I have no idea. Probably won't. But we're putting it in here just for the sake of, again, this is an academic exercise only. Okay. So we've got our SMAR in here now. And so the way that we did this was we cut with BAM-HI at the start to shove in this whole chunk, and then we'd be cutting with NDEI to shove in the SMAR. The other thing that we would put in after the SMAR, and this whole thing would be just not unsynthesizable, but, like, very, very expensive to synthesize. Jump to here just to get me to the end. So before that, now I've got to add the terminator. So all this is just going to get yeeted, basically. So the terminator we had, I don't know what happened to it. It should be in one of the other folders, probably in the glass turtle folder. Terminator, there we go, that's what we want. Cool, so we're going to just grab this whole thing, copy, go back over to this guy, which is the thing that we're working on. We found our spot where we need to grab, so that's where that goes. I don't need to worry about stop codons because this is not actual DNA. And then we paste our terminator. And that should just about do it. So I need to figure out where this actually ends. I mean, it ends here. So, yeah, 1883. So let's grab the annotation for this just to make this easier. 1883. Save. So now that should be a much smaller fragment and not look so janky. Let's grab this terminator. Figure where this starts. This starts at 3869. So now I'm just into the like adjusting annotations basically. I think that looks about right. No, it is not. Not even a little. I would. Okay, shit. What did I grab? 3869. Oh, that's why. Okay. Okay. So plasmid, but how do you deliver to the... Yeah, I fixed it. How do you deliver to the turtle egg? You drill a hole in the egg, and basically, you mix the DNA with something like PI or lipofectamine or something like that, and you just like inject into the egg, basically. The other way to do this would be, I mean, how large is this? Just from there to there, just to see if this. Oh no, wrong direction. Okay, length 3544. So you could do this as a virus actually, fairly easily, if you just stuck a. I don't know if AAVs work on turtles. You'd have to find the right turtle, the right virus. So that would be an entire level of difficulty, which I'm not prepared to get into right now. You could add, like, assuming we're trying to make this for humans, right? Like this, assuming this wasn't turtle, you would add like an AAV ITR to either end, and that would get, and so then you can use a virus to deliver it. But I mean, eh. But I think this means we're actually done. We have all of the stuff. This was definitely the most complicated thing that we've made on this stream so far. And it was hard, too. This was fun. This was a lot of fun. Such a ridiculous thing, but I love it. What is this? Empty. Oh, shit. I wasn't doing non-destructive edits. I was just going ham. Okay, we're going to call this glass turtle. So CD33 secretion, silica keratin fusion with SMAR. Okay. Cool. Very cool. Very, very, very cool. This is a really weird thing that we've made. I'm into it. Do you think the shell would really be like glass or mineral quartz? I don't actually know. I have no idea. I mean, the idea is that presumably as the turtle ate any like soluble silica that was floating around in its bloodstream that like passed over these cells, would stick to this protein and grow into like little nodules. So I think it would probably be closer to something like not a diatom. And it wouldn't be a nice smooth shell, I don't think. I think it'd be like little clusters, and you'd end up with areas where there was more silica or less silica. So I don't know, maybe like a mottled silica texture. I think it'd be kind of weird. But yeah, I think we've done the thing. So let's do a review of what we've done, and then I'm gonna do a couple minutes of questions, and then we're gonna wrap for the day. I think this was a very, very productive day. It was weird. I liked it. It was really nice change of pace from editing, which was, I appreciate. Okay, so let's close that so I can make this a little bigger. I'm going to actually make this like full screen so we can get this as big as physically possible. Wow, that made it way worse. Well, it kind of lets you see everything. And let's go one down. No, that makes it way worse. Okay, let's just keep it like this. Okay, so review time. So, what we wanted to, so the name of the game, the point of today's stream was to make a turtle express something that would make it more glass-like. Like the actual prompt just said silica turtle. So I took that to mean, what if we made the part of the shell of a turtle to be made out of glass? Just because I thought it would be really interesting. That seems like the most reasonable thing to do with silica turtle. Because somebody suggested, the actual suggestion that I got, and I think I got this through Twitter, was could you fuse a diatom with a turtle? Which we have actually done. I mean, we have no idea if this would work. It probably wouldn't. But, as an academic exercise, it was fun. So what we did was we first ended up finding we pulled up a paper that would analyzing proteins that happen to be expressed in the outer skin layer of a turtle's shell. So what we wanted a protein that would only be expressed there and not expressed anywhere else. So we found that. We found the protein. It's this. Don't actually know what it does. It's some sort of growth factor or something. And yeah, I don't know. It's kind of weird and interesting, but beyond that, it wasn't really what we were after. So what we ended up doing, and this is only a fragment. I don't know where the full, it might be this. If this is the big chunky boy, yeah, this is a big chunky boy. So this is where we got that from. So you'll see there's 3.8 million base pairs. So what we had to do was find in here, it's actually right here. It's this little fragment right here. Did it jump to it or did it freak out? Okay, yeah, it did jump to it. Cool. So we ended up finding this little piece in this huge assembly. And then what we did was we copied a section from before it and a section after it to give us our promoter and our terminator. And we even ran the promoter chunk through a tool that was supposed to find promoters, and it thinks that there's a promoter there. So seems like we did the thing. Cool. So we've got this chunk. We've got our promoter. We've got our terminator. Now, if we go back to glass turtle, which should be this guy, you'll see that we have our promoter, turtle shell promoter, and turtle shell terminator. It's right here. So now we've got something to flank the start and end of the thing. Very cool. So then past the turtle shell promoter, we're like, okay, well, we want this weird silica protein thing to be excreted out of those cells to make sure that the individual cells don't harden into little nodules. We really want a nice even silica layer on the outside of the turtle, which that sentence is the most ridiculous thing I've ever said. Moving on. So what we did was we grabbed a secretion peptide from mammals because there was no data on any sort of these things in reptiles. So we said, screw it. Let's just find something that should work. And so we went with a secretion peptide taken out of humans, I think, based on an immune cell secretion thing. And turtles do have a similar-ish immune system, so who knows, maybe this might work. I have no idea. Then we added a glycine linker. This is just going to be a string of, and I can actually show you this, literally a string of glycine. So actually, I'm going to make amino sequences of all of this just so you can see what's going on. Cool. So this is our secretion bit. This is our linker. This is the actual part that does the silica. So the RRIL on the end is the catalytic area. So this is the piece that actually accumulates the silica. Like this is what the silica atoms, or like the silicon, the silica, like the soluble silica will be attracted to and stick to. And once it sticks, it basically pulls it out of solution. Like if anything interacts with the silica, like it stops being dissolved. And also this peptide does that. So it pulls the silica out of solution, and then you end up with like a solid piece of essentially bioglass, which is really cool. This end is very sensitive to anything stuck to it. So I added another glycine linker just so that there's a nice space to make sure that it can function properly. Then to make sure that this behaves like the skin that we would want to have this expressed in, we also added some like a fragment of the keratin gene. In this case, we also used it from humans because I couldn't find it in turtles. You'll see it's very glycine rich. That's a very common thing with keratin and structural proteins. They tend to just be long strings of like glycine or alanine with like some other stuff like interspersing it. Primarily because the glycine is very crystalline, so it'll make the individual fibers stick together. So basically what this has done is we've got a silica thing and a thing that will make all of these individual proteins stick together really nicely. So it'll make like a nice smooth layer. In theory, I'm making this up as I go. We're making a glass turtle. There are no rules. Then I added another glycine linker because, I mean, who doesn't want more glycines? Everybody wants more glycines. And then I added another silica because why not? 2 for the price of 1. Maybe it'll work better. Maybe it won't. I have no idea. Again, I've added the glycine linker to the end because the end of this peptide is delicate, and you wouldn't want to offend its delicate sensibilities. Then we've got something that came with this starter backbone plasmid thing that I grabbed, which is Lactase. So in theory, you could stain the turtle with an enzyme, or not an enzyme, with a dye called XGAL, and any areas that are expressing this weird fusion protein thing should turn blue. So it's really an easy way of telling if the thing is working, which is helpful. And then finally, we added a stop codon to tell the ribosome, the thing that turns the RNA that this gets turned into protein, which is the thing that we want. It basically tells it that like this is the start, this is the end. But you will notice that there's this extra thing called SMAR. What SMAR does, and admittedly this is a human one, so God knows if it would work in turtles. As this thing is being read, like as this is being transcribed into RNA to get that process going, this area gets unwound, and then the proteins in the nucleus of the turtle's cells will bind this piece of DNA to the wall of the nucleus, making sure that it gets copied over during cell division, so that every time the cells divide, you continue to maintain this gene so that it doesn't like disappear after a round of division, basically. So you could like modify the turtle's egg with this thing, and in theory it should last throughout the entire turtle's life. Cool. That's that bit. And then finally we added the terminator, which is the thing that we talked about before. It's this actually makes the RNA polymerase let go. So it binds here, it lets go here, and everything in between ends up as RNA. Whereas of this huge long piece of RNA, the only thing that actually ends up as protein should be this part. There might be some issues where this is missing like a poly-A signal, like a polyadenosine signal, because sometimes RNA likes that too, but in theory it should be fine. It doesn't matter. And that's basically it. I say that's the whole thing that we designed, and I have no idea if this would work. I would probably bet that it wouldn't, but this is my most reasonable approximation for how you would make a glass turtle. That's part diatom and part turtle, because why not? So yeah, that was fun. That was a lot of fun. That was a lot of really cool bio stuff. Yeah, there's no way I could do this. You can't modify animals in Canada without like huge approval. It's a whole pain in the ass. This is why I work on yeast primarily and bacteria that don't care and the government doesn't care about. But This is a really interesting, weird piece of DNA. This is going to be uploaded to the GitHub, which I do not have open, but if you're interested in this weird bit of DNA, you can go to this repo. It's GitHub. Just look up whose gene is it anyway. All of the things that we've made previously, although I just noticed that I forgot to do the spicy tomato. So